I'm going to talk about Curio's spatial mapping kits, which is really changing the field, I think, in terms of the capabilities that are now possible with these kits. But just to introduce the company, for those of you who aren't familiar with us, we're founded in 2021, so relatively young company, only a few years old. It's formed by industry and academic leaders in the single-cell genomics and molecular counting space, including people such as Steve Fodor, who was the CEO and founder of Affymetrix a long time ago, is now our CEO, as well as Ari Cheney and Christina Fan, all of whom had sold a company called Cellular Research to BD and then eventually turned into Rhapsody. But our mission as a company is to advance a new generation of high precision tools for the spatial biology market. And we want to provide our customers with a disruptive technology or technologies that are easily accessible and can accelerate everybody's scientific discovery. So as a bit of an introduction into spatial, I'm sure many of you are very familiar with various aspects of this space, but there are really two categories of spatial platforms that are out there now. They're the fish-based platforms and the sequencing-based platforms. So a lot of these platforms can be very difficult to run. They're very cumbersome. And as some of you may know, it requires, or some of them require very, very expensive capital equipment. So it's very difficult right now to get into spatial. Additionally, there are some perceived trade-offs between the resolution and number of targets, depending on which platform you choose. And so really what Curio is trying to do is address all of these concerns with our platforms. And our platforms are based off our core technology, which are 10 micron beads that are placed in a very closely packed monolayer on a tile, as we call it. It's in the shape of a square. And each of those beads have a ton of oligos on them, each with a PCR handle, a bead barcode or spatial barcode, a UMI, and a poly-DT to capture any RNA that have poly-A tails. And so now we have two platforms. First is the Curio Seeker spatial mapping kit, which is based off of SlideSeq. And more recently, as we announced last week, we have entered early access for Curio Tracker, which is the first truly single-cell mapping kit, which is based off of slide tags. And so first I'll be focusing on Curio Seeker for the first about half of the talk, and then I'll move into Tracker later on. So Curio Seeker is a high-resolution whole-transcriptome mapping kit based off of SlideSeq, as I mentioned earlier. And all you really do is you section a 10-micron section of fresh frozen tissue. You melt it on top of our tile of 10-micron beads. Come in two sizes. There's a 3x3 tile and a 10x10 tile. The 3x3 tile has about 80,000 beads on it, so you have the potential to get about 80,000 individual transcriptomes. And similarly, the 10x10 tile is much larger format with about 800,000 beads. Once you melt the tissue on top of the tile, you'll just immediately put it into either an Eppendorf tube or a custom injection molded adapter that we have with hybridization buffer, depending on the tile size. And once you have it in hybridization buffer in the tube, you'll just go through an RT step, a second strand step, all within the tube. You'll take the beads off, go through wash steps, and then very typical basic molecular biology techniques to generate a sequencing library. And once you get to a sequencing library, you can sequence your libraries and then put it through our pipeline, and you'll eventually get a map of your tissue and the differential expressed genes layered on top of it. And it's a really easy protocol, right? There's no additional equipment that you need. You can get from your tissue section to a sequenceable library in about 8 hours. And it's just very common lab equipment, just heat block, PCR machine, Eppendorf tubes, pipette tips, very common things that you see lying around in the lab. And what I'd like to emphasize here is that in this protocol, it's fresh frozen tissue and there's no fixation or permeabilization. So there's absolutely no pre-optimization required for this. It's the same exact protocol for all tissues and all species. So we've had people running this on, non-model organisms like cuttlefish or octopi or planaria, just all types of species. And for tissues within the human body or mouse tissue, it's the same protocol. So it's very, very simple protocol that anybody can implement. And so once you generate your data, this is the type of data that you can expect. So here's a mouse hippocampus data set, which is very common in this space because it's very easy to see structure within the tissue, but I'll show you examples of other tissue types in the following slides. But you can see it's much higher resolution than you can expect from competitors, right? You can very clearly see structure within the tissue, and you get really good capture per bead. Obviously, it will vary where you are within the tissue, what the capture efficiency or rate is going to be, depending on if there are actually cell bodies in that area or not. So for example, where there are a lot of axons or non-cell body areas within this tissue, you're not going to see very much capture because there's nothing there. And so you get a very good broad capture of transcripts across the tissue section that's on the tile. Once you put it through a pipeline, you'll get several different file types. There's a HTML report file that includes the image that you just saw in the previous slide, as well as an unbiased UMAP of the clusters in your tissue, and then a spatial map of where all those clusters map to, as well as a bubble plot of different genes that drive each cluster, and the top 10 differentially expressed genes for your tissue type. And so I'll show those images in the following slides. But a lot of the other files are files that you can use for any secondary analysis that you want to do. So very common prepackaged files you'll see here. There's a Seurat file for people that want to use Seurat, which is a very typical single cell analysis tool, also used for spatial. And then there's a ScanPy file, the H580 file, and also just generic raw matrix files for use in whatever pipeline you want to use. So it's very, very flexible for secondary analysis. These are just some metrics that are typical on the HTML report. So in the top part of the report, metrics table, you'll see some metrics that just tell you how deeply you sequence. Have I reached saturation? How many what percentage of adapters are in my library? That will indicate how clean your library is. So if you see a higher number, typically that means you just didn't clean your library well enough, but it's a good measure of how your experiment went in general. Next, you'll see a few statistics for how many beads are recovered. This typically is dependent on not only bioinformatically how many are recovered, but also how much of the tile is covered by tissue. So if only half of your tile is covered by tissue, maximally you should expect 50% of barcodes recovered, but you don't really see that. So percentage-wise of everything that's there, we see anywhere between 70 and 95% of beads recovered. So that's relatively expected. And then there are two filters below that. So how many of those reads match to barcodes from the whitelist for every tile, because every tile is unique, and how much match the barcodes and the genome reference. And so this percentage is typically slightly lower because you're going to be collapsing multi-mapped reads or the like. And so this number is usually representative of what you have. The last three statistics just tells you how high quality or low quality your tissue was. So if it's low quality, these numbers or percentages typically are higher. But if your tissue is of very high quality, you see anything below 5% or 10% is usually very good. There are exceptions to this. So for highly active tissues that have a lot of transcription or translation, you'll see higher numbers for ribosomal protein or RNA. But generally, these are pretty low for good samples. In the HTML file, you'll also see these two figures. On the left, you'll see the unbiased UMAP clustering, where you'll see all the different populations of cells. And then on the right, you'll see where all those populations map to. You can see very nice structure within the tissue. And here you'll see all the genes that are driving each cluster in the map that you just saw. So here you can see very nice definition between the clusters. And these are just the top spatially variable features within the data set. And here you're just looking for basic structure within a tissue, which will tell you whether or not the assay worked pretty well. And usually, for even tissue types that don't normally have much structure, just looking at it like tumor tissue, usually you can see very nice structure here already. And I'll show you some of those data sets later. As I mentioned before, this works on all tissues and all species. So here's just an example of mouse liver with very nice structure around the portal vein. And here is human breast cancer with a lot of genes here that are indicative of cancer as well, or breast cancer, as well as immune cells in the bottom right, which you would expect to infiltrate a cancer in most cases. And so for a more clinically relevant sample, we have a human melanoma sample where here you can see nice structure. You're not quite sure just looking at this what they are actually, but if you look at the bubble plot, you can see some of the clusters are specific to tumor cells or immune cells or epithelial mesenchymal transition cells, as well as normal skin cells. So if you look at these specific populations in different colors grouped together, you can nicely see that immune cells are very nicely infiltrating the melanoma cells in the upper left-hand corner, and they're appearing exactly where there's no melanoma cells, which is very cool to see. And then on the bottom right-hand corner, you have the conversion zone where all the EMT cells are, and then the differentiated markers for normal skin cells. So even just basic, structural observation here, you can see very nice functional, or draw a lot of functional conclusions here. Now, because of the small bead size or high resolution here, you can see that for immune markers, you can get very high resolution depictions of your immune cells. So you can see individual immune cells showing up as individual beads, which you don't normally see in a lot of other platforms. Obviously, for cells that are slightly bigger, like glial cells that are typically 20 to 30 microns in size, you'll see clumps of 2 to 3 beads. But here, it's really nice to see that you can see individual cells. If you want an example of a paper that has mostly seeker data in it, this is a more recent paper that was published late last year in December from Cornell out of Ewan de Vlaming's lab, where he was looking at the kinetics of changes in the ovaries over time after stimulation. And so the study was designed such that you stimulated ovaries, collected samples over time, and then mounted it on our 10 by 10 tile. So he was able to mount multiple ovaries in the same tissue block, section it once, melt it on top of the tile, and then run the Curio Seeker assay all the way through to observe the kinetics and spatial organization within the ovaries. And this is basically what he saw, right? There are very clear differences between each time point, as you would expect, and there's a lot of progression in the changes in the different cell types and populations of cells over time. And so I'm not going to go into this in any depth, really. There is a webinar on our website where he gives a good summary and presentation on what he was trying to find here. And you can also read the paper if you like. Evan McCosco's lab last year published a paper where he was looking at or trying to reconstruct a 3D model of the mouse brain, the whole mouse brain. And he did this by sectioning over 100 slides in a couple of days and getting to FASTQ files after sequencing to reconstruct this. Now, the point of this is that with the Seeker platform, it's very amenable to high throughput studies like this, where you need a lot of data within a short amount of time to reconstruct whatever you're trying to reconstruct, right? So you can get this type of data in a very short amount of time within weeks, whereas in other platforms and fish-based platforms or others, it can take months sometimes because the runtime for each section can take very, very, very long time. So that's another strength of this assay. I'm just going to go through very quickly some high resolution data for the larger format, which is the 10 by 10 millimeter tile. Same data type, same high resolution, just a larger format, as you can see here. The inset here is approximately what a 3x3 tile area would look like, but obviously you have a much larger area now. And again, much higher resolution than what's out there currently, and with very comparable capture efficiencies to what you saw on the 3x3 tile. Again, here's just an unbiased UMAP of your tissue, in this case, the mouse brain, and where all those populations are mapping to within the tissue. And then if you look at individual genes, again, you can see very nicely there's very high resolution structure within the tissue. So by this point, you should be confident that you can dig into the data more deeply and find the genes that you might be looking for. Here's another example for the 10 by 10. This time, instead of taking a small piece of the melanoma, now we're looking at the full melanoma tissue. And here you can see some, very minor structures within the tissue here, just based off the total UMI capture efficiency. But once you start clustering everything, this is where everything starts getting exciting. We can see a lot of structure within the tissue, and you really want to see what those clusters and cell populations are. If you look at the top 10 differentially expressed genes, not surprisingly, you see a lot of immune cells here that are in the tissue and infiltrating the tumor. If you look at genes specific for melanoma here, you can see that the melanoma is actually on the surface in the upper left-hand corner. And more recently, in the last few years, there were a couple more genes that were identified as being important for progression of melanoma. And so the point being here is that if you just in one experiment, you can not only see the markers that have been identified over the years for being important in identifying melanoma, but also new discoveries that have been made in recent years. So basically summarizing several years of experimentation in one experiment. So this is a very, very powerful discovery tool where you can use this to discover new insights into whatever you're looking into, and then validate that with commonly used assays like FISH or antibody staining, what have you. And this is just an example of a very pretty picture. So obviously, mouse embryo has a lot of nice structure within the tissue. And so we just ran this recently for mouse embryo and got really nice data. And again, you can see very high resolution structures within the embryo itself. Okay, so this is just a summary of Curio Seeker before I move into Trekker. But everything here I've discussed except for the sequencing depth. So for the 3x3 tile, if a tissue is covering the whole tile, we usually expect you to probably sequence in the range of 500 to 600 million reads to get to a decent amount of saturation. Obviously, people would choose to do more or less depending on what they're looking at, but that's really up to you. But 500 is about is the average. For the 10 by 10 tile, just because there's a ton of beads on there, this moves up to about 2 billion reads, which is very typical for various tissues, but that's if it's covering the whole tile. So if it's usually covering half the tile, you could expect maybe 1 billion reads for the full for the tile. Any questions before I move on to Trekker? If not, I'll head along. Okay, so I'm sure many of you have been in the single cell space for quite some time. Single cell data is very powerful, and you can work out all the different populations of cells and what's important for your tissue type. But obviously, the question for many people in the last several years is where is everything in the tissue? You don't really know where your cells are. There's a lot of biological importance that is there, but has not been unveiled in single cell RNA sequencing data alone. So that's why a lot of people have been going to spatial platforms in the last few years. And as with any industry, there's been a huge race to platforms that have higher resolution, right? So they all want to get to high resolution because the thought is that if you can see at a higher resolution, then you should be able to see whatever you need to see within individual cells. So across the board, across the different platforms, there's quite a range of different resolution where you go from maybe 50 to 100 cells all the way down to subcellular resolution with the fish-based platforms. But the reality here is that everybody's trying to get to the same answer here, which is single cell resolution. So the only way that these platforms can achieve this or try to achieve this is through either deconvolution or segmentation and deconvolution and integration in the end, right? But as you may know, this is very difficult because, for deconvolution, there's a ton of different pipelines that are used for this. There's not really one well-accepted pipeline that everybody uses because they all have their pros and cons. And so it's very difficult for people to figure out which one they want to use. Similarly, with segmentation, here's a very pretty picture of a tissue, but you can appreciate here that it's very, very difficult to segment the cells here. You'll never be 100% correct. Even in tissues that are more amenable to segmentation that look easier, you're still never going to be able to 100% trust that what you're segmenting is actually real. And this is reflected in the UMaps that you typically get from these platforms, including Curio Seeker, where you see a lot of overlap between the clusters, right? And this is just a result of the fact that you can't accurately segment the cells or deconvolute them into actual single cell resolution. This obviously is different for single nuclear RNA sequencing data, which is generally cleaner because you have much more distinct entities in your nuclei that can separate out more easily. So it'd be really nice if you could somehow combine these two different worlds into one. And that's exactly what was done with slide tags, which was published late last year in December as well. And this is the first really true single cell spatial solution that's out there. And the reason that you don't have to deconvolute with single cell data is because it actually is truly natively single cell data already. So you don't have to do any transformations, you don't have to segment, you don't have to deconvolute or anything. And so this is just taken straight out of the paper, but basically what you do is you section a 20 micron section, put it onto the same type of tile with the 10 micron beads, except this time the oligos that are on the beads have a photocleavable linker that can get released when you expose it to UV light. So once you expose the tile plus a tissue to UV light, the oligos get released, and then they go into the tissue and the nuclei. And so what happens is that once you isolate the nuclei, they all have their own spatial tags, and then they go through a single cell platform such as 10x, and then you can sequence your data. And so in the end, what you get is a collection of nuclei that you can cluster, as you're very familiar doing already. But each of these data points or nuclei have their own collection of spatial barcodes from the tile. And so you can imagine that each nucleus will have a different combination of spatial barcodes depending on how close or far it was away from any particular bead within the tile. And so what you can do is use that collection or proportion of different spatial barcodes for any given nucleus and triangulate where that nucleus originally was to within about 2 to 3 microns of the original position. And this accuracy is afforded because you have information from multiple beads on the tile. And so Evan McCosco, who is one of the inventors of slide tags, recently presented some data from postmortem human cortex tissue where he sequenced 26,000 nuclei. And you can see very nicely already the different layers of the human cortex. And actually, after integrating with the annotated single cell Allen Brain Institute data set, when you look at the different cellular or cell types within the tissue, they show up exactly where you would expect them to. And then with the excitatory neurons specifically, they obviously have a distribution or known distribution of within the cortex, and you can see it very clearly here. But was unexpected and was new for the researchers was that the oligodendrocyte precursors, which had no known spatial distribution within the tissue, suddenly they showed a nice spatial distribution. And so if you look at the representative genes within this population, you can see that some of them show up in the deeper layers of the cortex and others do not. And so the point of this is that single nuclei RNA sequencing data alone, these precursors cluster is just one cluster because there's not enough variance within the tissue or within the cells to pull them apart. Whereas it's only after you add the spatial component do you actually see this new pattern that had not been shown before. And this has really been the promise of spatial for several years now, but depending on the platform, it hasn't really realized its full potential because of some of the limitations that I mentioned earlier. And so this can be due to limited panel sizes because you're not looking at enough genes, or this inability to 100% isolate individual cells. And so this is the first time you can pretty much 100% trust what you see from this because the data is just natively single cell. And this is our product tracker, which is based off of what I just showed you for slide tags. And it's the same protocol. I use a 20 micron section of fresh frozen tissue on there. And we have that photocleavable linker, expose it to UV light for about a minute. And then you isolate your nuclei and put it onto the 10x platform. So currently the handshake sequence that we have on our current version of the beads is complementary to the 10x feature barcode 1 sequence. And so what you do is once you put it through the 3' gene expression assay with the feature barcoding, you get two libraries, one of which is a gene expression library and the other one which is the feature barcoding or tracker library, as we call it. Once you sequence both of these libraries, you'll get FASTQ files for both of them. And then you'll you'll put the FASTQ files for the 10x gene expression library through the 10x pipeline to get the output files. And then you'll combine those output files with the tracker library FASTQ files through our pipeline. It'll just spit out all the locations of the nuclei.
Excuse me, Bertrand, but there's a question in the question and answer. Mind, how easy is it to triangulate the nuclei back to its location, and what bioinformatics tools are needed to do this?
Yeah, this is actually very straightforward. We build it into the pipeline that we will be providing on our website. It's not up there yet, but we're just finalizing that right now. But it's very simple. And so we've built it in. You don't have to worry about it. But it's essentially triangulation using the different barcodes, calculating what proportion or one barcode versus another. There's going to be several in a cloud of barcodes around a nucleus. And so depending on that cloud of barcodes, it's very accurate to position that nucleus. I hope that answered the question.
Okay, oh, we've got another one. What's the analysis? Sorry, what the...
Oh, is it analysis influenced by the shape of the nuclei?
Yeah, nuclei are elongated instead of round. Will that affect the analysis?
Yeah, I guess it would somewhat. I suppose it's still pretty accurate because let's assume it's very long. You know, the ends would get nuclei, or sorry, spatial barcodes associated with it from the extreme ends, whereas the inner parts of the nucleus would not. And so just by that ratio of close or not close to it, it should position it still pretty accurately because it's going to get collection from around the beads around it, depending on how far close it is. So I would say that the positioning should still be pretty accurate. Yeah, just like, you know, in GPS, if you have 10 cars in a line, you should be able to position, you should be able to tell where each car is within that line, and then taken together, you should be able to see where that line is within a map, or yeah, in a map. So I think it's the same concept here. Okay. Yes, we're working on it, but there's no timeline for when that will be available.
Sorry, just for everyone else, that's answering the question about compatible with FFPE samples.
Right.
So the idea is we're working on it. At the moment, it's only compatible with fresh frozen, frozen samples.
Yep. Yep. Okay. I'll go on for now. So the next question I usually get for people that are interested in this is how many nuclei should I sequence? Because you're isolating a bunch of nuclei, but it's really up to you how many you need to sequence, right? So I usually ask people how many nuclei they typically isolate from any given tissue, and the answer is somewhere in between 500,000 and several million. And this is very typical for nuclei and for cells in general, because you're just taking a chunk of tissue and you're trying to get as many cells or nuclei out of it, and then you're just going to load 10,000 nuclei out of that, because that's usually all you need for a decent single cell experiment, right? Just to identify the different cell populations and annotate them. If you put and layer a spatial component on top of that, just percentage-wise, this is actually extremely sparse data, right? So if you're isolating 5 million cells and only isolating 10,000, your data looks very sparse, right? This is just actually a subsample data set from one of our current tracker data sets, and you can't really make out what it is because it's just too sparse. Even if you go up to 2%, you can make out the border of the tissue, but you can't really see what's inside of it. What's nice is that even when you get to 10 or 15%, you can already nicely see a lot of the structures within the tissue. So you can kind of make out or actually you can pretty clearly make out the hippocampus here and the different layers of the cortex, as well as other structures within the tissue, just with 50% of the nuclei. And what's nice here is that from a typical mouse brain section, we get anywhere between 50 and 150,000 nuclei out of that. So by default, even though you're only loading 10,000, you're already getting a much higher percentage because your starting material is so low, because you're only taking a 20 micron section, right? So at first, when you tell somebody 10 or 15%, that seems pretty low, just mentally. When you see the actual data, is actually probably enough in most cases. I would say that if you are looking for finer structures within the tissue, you probably have to sequence more nuclei. And then obviously, if you're looking for more gross structures or larger structures within the tissue, it's less necessary to do that. And so this is just kind of a summary of what I just said, but mouse brain, we typically get 50 to 150,000 nuclei. I was at a beta test site that did human liver on the same size tile, which is a 10 by 10 millimeter tile. We got about 40 to 50,000 nuclei there, which was probably undercounted because we were doing it by hand. But that's in the range of what you should expect for a tissue that covers maybe 3/4 of the tile. As I mentioned before, you can sequence more nuclei if you want to. This is an example of a mouse embryo again, where we recovered about 130,000 nuclei, and we just ran everything. And so plotted here is about 83,000 nuclei. So looks very nice. You can see a lot of the finer structures within the tissue, but you by no means have to do this. There are probably more efficient ways of looking at your data, which I'll discuss later. Just a sampling of the bubble plot. So this is very similar to Seeker, but essentially what you're seeing here is very well-defined gene sets that define each cluster, and is very similar to normal single nuclei RNA sequencing data. It's actually the same data quality. It's just that you have a spatial component now, which is very nice, right? So you don't lose anything by doing this. And if you look at individual genes that define different cell types, you can see them showing up exactly where they should be showing up within the tissue. So we do have two platforms now. We have a tracker and Seeker. We do see them as being complementary in the discovery space. So it really will depend on your scientific question, right? So Seeker provides a more continuous view at a single cell level, a map of your tissue. And so there are no holes in the data. You're seeing everything across the tissue. But it's maybe not quite as deep in terms of the gene coverage as you would expect with Trekker, which provides not only true single cell resolution, but provides you with a much deeper picture of your gene coverage, right? And so you see a lot more genes with single cell data in general than any spatial platform out there right now. And of course, it's scalable for the amount of sequencing that you do. And some people are thinking of using both in tandem with each other. So, Trekker data is actual single cell data, and you could possibly use it to deconvolute Seeker data because it's more sparse. And so they work very, very well with each other. And so you can imagine using Trekker data and Seeker data from the same exact tissue block, which is a lot more powerful than using some random single cell reference to deconvolute your spatial data. And so you get data from the exact same block, and you get the best of both worlds. And so just with the general recap of the benefits: true single cell resolutions, whole transcriptome by leveraging single cell platforms. It's only a 1 hour protocol upstream of the single cell platform, so it only takes 1 hour to isolate your nuclei. And a nice side benefit is now it preserves or helps preserve your tissue, because you're only taking one 20 micron section of your tissue block as opposed to a big chunk where you will isolate a ton of nuclei and then throw away the rest of them. Right? So now you can preserve it for any other assays you want to run on that tissue. And generally, it makes it extremely easy for any single cell scientist to get started in spatial. So I would imagine there are a lot of single cell scientists out there right now who are kind of on the sidelines because the spatial space is very crowded, very expensive, and there's no consensus as to what's the best platform to use. And so this makes it very easy to get a very early answer to what's going on in your tissue just by adding this 1 hour protocol upstream of single cell assay that you're already running. And so this is just a summary of what I talked about. 1 hour workflow, works with the 10x Chromium 3' kit with feature barcoding. And it's the first true single cell resolution product on the market. So that is it. Anybody have any questions or additional questions?
I think it's good to note that the tile size has to be for the Trekker product needs to be 10 millimeter.
Yes.
Yeah, you can't do it on the 3 millimeter ones.
Yeah, 3 millimeter would be too small because you would not recover enough nuclei to put on a single cell platform. We are exploring smaller tiles, maybe something in the range of 6 by 6 or something. But we'll see what we determine to be needed by the space. So
okay.
Yeah.
I have a question. I'm sorry if this sounds a bit silly, but does that mean if you want to do Trekker and Seeker in the same block, you actually need to take 2 slides or 2 slices? Right.
Yeah, the sequential section, yes. Mm-hmm. Mm-hmm.
Anyone else? Sorry, does anyone else have any other questions?
Wish I could allow people to talk, but probably not. Okay.
Nope, no problems. We'll share the webinar quite widely. So for anyone who's missed it, we can share the webinar for them. If you have any further questions at all, by all means, probably best to contact me. Bertrand's a bit busy, but anything that I can't answer, I will obviously pass on to Bertrand as well.
Okay. Cool. Well, thank you, guys for joining. And thank you, Catherine, for organizing this.
That's right. It's fine. We'll be in touch. So I will be in touch actually just a bit later with everyone who's attended, to see if we can get your thoughts on the platform and to see if you're interested in trialing it. In the meantime, thanks everyone.
Okay. Thank you, guys.
Have a great day.
You too. Bye-bye.
Thanks, Bertrand.